Everybody can hate her now, okay? So, all right, so we have a fair number of things to get through. I'm going to finish talking about protein isolation techniques, and I'm going to talk about an isolation technique that most people, when they see it, go, wow, okay? Because you can start to see how it is that isolation, which may seem like it's kind of mundane, all of a sudden shows you things that you might be interested in with respect to human health and so forth, okay? So before I do that, I need to mention a couple of things. I talked to you about how we separated proteins. And I told you we had to use some tricks to separate proteins. We had to make the protein, make it long, that is, unfold it, and we had to coat it with something that gave it a negative charge. That was the SDS, okay? That gave it the property that it was like DNA. It was a polymer of negative charges, or at least as far as the gel looked like, it looked like that. All right. Now I need to tell you about another technique, but it's kind of a complicated technique, so I'm going to give you just the headline about it. And if you're curious and you want to know how it works, come let me know, and I'll be happy to tell you. However, I won't require you to know how it works. Okay? Is that a deal? Oh, by the way, we have an exam a week from today. Did you guys know that? Oh, my God. That's the right response right here. We have an exam a week from today. You guys all ready for that? Question? Will it cover material up through next Wednesday? I haven't decided where that'll cut off. I usually announce that once I get to a certain point. Usually it'll cover through enzymes. And I'm getting ready to talk about enzymes today. So I will let you know. Yeah? How are my tests formatted? Okay. So for my classroom class, which is what you guys are, I provide practice exam. Not a practice exam, but an old exam that you can look at it, and you can see the format. And you should look at that. I'll post that this weekend. Okay? eCampus, I don't do that, and I don't do that because eCampus takes multiple choice exams. I don't encourage anybody to use old exams as practice or study guide because things change from year to year. The reason I don't give it to the eCampus is because they already know the format. It's multiple choice. This class does not use a multiple choice. It uses a different format, and I want you to see the format before you take it, okay? But I really strongly discourage you from using exams as study guides or anything like that. Your own study approach is much better than an old exam, okay? But I will provide that for you this weekend. Okay, and I'll also, yes, I do review sessions, and I'll do a review session for you next week sometime as well, okay? Where you can ask questions and all kinds of stuff. All right, well, I got sidetracked. I'm going to come back to the subject. And the subject is I need to tell you about another technique because what we're going to do is we're going to combine two techniques into one. And when we combine those two techniques into one, you get something that's really amazing. Really, really amazing. Okay? Well, the second technique I need to tell you about is something called isoelectric focusing. Okay? Isoelectric focusing. So first I'm going to tell you what the technique is, what it does, and then I'm going to tell you how we combine it with the other technique to do something pretty amazing. Isoelectric focusing. Now, I said that isoelectric focusing is kind of techy, so I'm not going to expect you to know how isoelectric focusing works. But I will tell you this. Isoelectric focusing separates proteins on the basis of their charge. It separates proteins on the basis of their charge. You've already seen proteins can have different charges. And what isoelectric focusing does is it separates proteins on the basis of their charge. Okay? You see this here, it says pH 4, pH 10. Don't worry about the pHs. That's the techie part of what's here. Suffice it to say that the proteins on the left will be the most negative, and the proteins on the right will be the most positive. And you don't even need to know that. All you need to know is I have a spectrum. I separate a bunch of proteins out. The middle of the gel that I run has proteins charged 0. One direction away from that 0, I get a protein charged +1. The other direction away from that 0, I get proteins charged a -1. -2, +2, -3, +3. Everybody understand? So that if I were to go from the bottom of this guy to the top, I would be looking at something like -12, -11, -10, -8, -7, blah, 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 all the way up to 0. And then it would go +1, +2, +3, +4, +5, et cetera, et cetera. Everybody understand that? The ends will have the most positive or negative charge, and the middle will have a charge of 0. If I said, tell me what you get in an isoelectric focusing gel, you could presumably tell me what I just told you? The correct answer is yes. Okay. All right. Anybody have questions about that? Yeah. The point I want you to hammer in is that this technique separates on the basis of charge. The most charged things will be at the ends. One end will be very positive, the other end will be very negative, and the middle will have a charge of exactly 0. Everybody understand that? Now we're going to combine the two techniques, and you go, oh my God, you make it complicated. It actually isn't complicated, but the result is really amazing. Here's what we're going to do. We're going to take our mixture of proteins that we've busted open cells, and we've got these hundreds or thousands of proteins that are sitting there from these cells that we've busted open. The first thing that we're going to do is we're going to do isoelectric focusing on those proteins. We're going to make a gel that's going to look like what you see here. Now, these proteins, because of the way we use this technique, won't have negative charge because we're doing something different, right? I told you it's a techie thing, so we're not coating them with SDS or anything like that. They're in their native structure, their native charge. Everybody understand that? So the protein starts out with a certain charge. We haven't done anything to that in this, what I've described so far. The proteins that are charged 0 will be in the middle. Proteins charged very positively up there. The proteins charged very negatively down here. Okay? I've got those guys separate. I've got this slither of a gel, and now I take this slither of a gel and I lay it on top of another gel. So you can see I've taken this, I've laid it sideways across the top, and now I add SDS. Why would I add SDS? Unfold it and make it negatively charged so I can do SDS-page for the next phase. The next phase, I've got the SDS there, and now I separate going downwards on the basis of size. So if we think about this, left and right, I have separated on the basis of charge. Up and down, I've separated on the basis of size. It's called two-dimensional gel electrophoresis. Two-dimensional gel electrophoresis. I'm going to tell you how powerful it is in just a second. Okay? When I have finished this process, you can see that I have spots all over this gel. That spot right there, you know, is going to be big, because big guys go slow, and big is going to be near the top. This guy is going to be big and positively charged. This guy down here is going to be small and negatively charged. This guy right here is going to be 0 charge and about medium size. Everybody see that? The beauty of this technique is that I can take a mixture of cells and I can separate every protein that's in there, and every protein will show up as a spot, just like this. But now I can look at this and say, big, positive, big, negative, medium, 0, etc. Well, who cares? I'll tell you who cares. I can use some really sophisticated techniques and determine what every one of those proteins is in the cell and what it does. But it's better than that. Okay? It's better than that. Let's imagine that I have a tumor. I have a tumor on my liver. Okay? And my doctor is interested in knowing what proteins are present that are causing this tumor, or what proteins are absent that are causing this tumor. Here's what the doctor would do. The doctor would take that tumor, bust open the cells, do 2D gel electrophoresis, and the doctor would take a part of my uncancerous liver and do the same thing. I would have 2 gels. One gel is of non-cancerous proteins. The other gel is of cancerous proteins. I compare the two. Some are going to be proteins in both. Some, I'm going to see a spot in one, but not in the other. Do you suppose that would be of interest to my doctor? You betcha. 2D gel electrophoresis allows a person to look immediately and tell the difference between 2 conditions. Cancer versus non-cancer? Maybe I wonder what a drug does. Maybe I think that a drug that I'm giving a person is causing some problem in their lungs. I take tissue from a person who isn't taking the drug, compare it to the pattern of a person who is taking the drug, and I can ask the question, what are the differences? And since I know what each protein is, I know what is the actual effect of that drug, or what is the actual effect of that cancer. 2D gel electrophoresis is phenomenal. Question?
Will they ever blend together, or will it be like 2 proteins and it's like
I'm not sure I understand the question.
So, will it ever blend together?
Will 2 proteins ever blend together? Generally not. This technique is usually done on a pretty big gel, and the separation technique is good enough that every individual protein will get separated. They generally will not blend together. That's pretty cool. Yeah?
On the step where you have the protein downstream and then you add the SDS-page,
Yes?
because you said it's separate from charge like inside, but when you add that to the protein, it's negative, but they lose more protein.
Okay, so here's a good question. All right? So I said I've got these proteins up here. They're separated on the basis of charge, and then I add SDS to it. Isn't that SDS now going to make these guys all negative? Aren't they all going to go one direction or another? They're in a gel. They've already been separated. It's a very, very good question, though. They've already been separated, so they won't move any further, because what caused them to move in the first place? Electricity. And so I don't have electricity going left and right on this gel. Okay? Very good question. Other questions? Pretty cool stuff. All right? This is one of the most powerful tools that we've invented for what's called proteomics. And I'll spell that for you. Proteomics. One of the most powerful tools for proteomics we have invented, okay, in the past 15 years. Very, very powerful technique. Okay. So, knowing how to separate proteins allows you to do some pretty amazing things. I'm going to mention a couple of other things very briefly, and then we're going to start talking about enzymes. The last technique I'm going to give you for separating proteins is a technique called HPLC. And yes, you can know it as HPLC. You don't need to know what it stands for, but I'll tell you. It stands for High Performance Liquid Chromatography, blah, blah. Please turn the phone off. Okay? High Performance Liquid Chromatography. High Performance Liquid Chromatography is a technique, some people call it high pressure liquid chromatography, because it uses columns like you've seen before, except they're packed with very, very microscopic beads, very, very tightly. And the solvent that passes through them, the buffer that passes through them, okay, has to be pumped through at very high pressure because it's packed so tightly. Now, I'm not going to go through the details by how this works, but I will tell you what the separation basis is. This technique separates on the basis of polarity. Polarity. Polar versus nonpolar. That can be very helpful for us if we're talking about amino acids, for example, that are polar versus nonpolar. Things in this technique, okay, will separate on the basis of polarity. Now, this is a special technique that's set up here that unfortunately is not set up to do on the basis of polarity. It's using another process. But the HPLC technique in general separates on the basis of polarity. Okay? In general, a nonpolar molecule will come out last, and a polar molecule will come out first. Nonpolar last, polar first. Okay. Let's talk about one other thing, and that's actually something that we have to do to proteins sometimes to study them. Sometimes, in order to study proteins, we have to break them into smaller pieces. Proteins can be rather large, and large pieces can be unmanageable for some things that we're trying to do. It's therefore desirable that we have ways of breaking proteins at specific places within the protein. Specific places within the protein. Okay? These things that you see on the screen, and I'm going to show them to you in a second, cut proteins at specific places. Trypsin is an enzyme. Trypsin is specifically a protease. A protease is an enzyme that cuts another protein. A protease is an enzyme that cuts another protein. And it cuts it at a specific place in that protein. In the case of trypsin, trypsin cuts on the carboxyl side of arginine and lysine. Wherever a protein has arginine or lysine, trypsin will cut it right there. Should I memorize that? Oh, it wouldn't hurt. Okay? There's one thing I'm going to have you memorize about stuff, right? Trypsin's cutting on the carboxyl side of arginine and lysine. Okay? You can see if I took a protein like this down below and I cut it with trypsin, every place there was an arginine or a lysine, I would cut it accordingly. Everybody follow? Okay. Chymotrypsin is another protease that will cut things, and I'm not going to expect you to memorize what chymotrypsin cuts at, because it cuts at several amino acids. But the point is, chymotrypsin will also cut proteins. These are proteins from our digestive system. Our digestive system uses them to break down the proteins that we eat, so that those amino acids can be taken and used to make new proteins. Okay? The reason I mentioned chymotrypsin, why do you even mention it, is because we're going to talk about chymotrypsin a little bit later in the term, and I just wanted to expose you to it. Okay. The last reagent that I'll talk about, this is called a reagent, it's not an enzyme. This is a chemical that cuts proteins. And this chemical cuts proteins specifically at methionines. Methionines. Okay? Here's a methionine. Here's the reagent. It's called cyanogen bromide. Sorry, I can't even get the word out. All right, it's called cyanogen bromide. There's cyanogen bromide right there, and you can see it cutting specifically at that point. Now, those tools for cutting proteins at those specific places we will talk about later, but they're very, very valuable tools. And the reason that they're so valuable is because they cut at places that are only those places. They won't cut at other places in the protein. Okay. We'll come back to that. Yes, question? Cyanogen bromide cuts at methionine. That's right. Yes? Okay, it's a good question. So it doesn't. So these things will cut on a specific side. You're exactly right. The side, if we look at how we write this protein, you can see that there's an amine side and there's a carboxyl side. So the ones that I've described to you will all cut on the carboxyl side of a protein. Okay? Now, that for most purposes doesn't matter very strongly, but it matters when we get to the end. Let's say that I had, at the very end, I had a methionine. On the carboxyl side, there's nothing to cut off. On the amine side, if it cut on the amine side, what it would do is it would cut methionine off. Make sense? Yeah. So the side matters only when we get to the very ends. Other questions? All right, we're going to talk about some fun stuff now. Yeah. All right, let's talk about enzymes. Let's talk about enzymes. Enzymes are incredible. All right? I'm going to show you a couple techie things about enzymes, and then I'm going to tell you why enzymes are absolutely remarkable. Okay? Here's a techie thing. Okay? It shows what enzymes do. Now, why do I even care about what enzymes do? All right? Here's why I care what enzymes do. I told you in here earlier how fast enzymes work, right? Or not? Have we talked about how fast, how many that they... Yeah. Okay? An enzyme can speed up a reaction 180 quadrillion times. A chemical catalyst can speed up a reaction 1,000 times. 180 quadrillion is a hell of a lot more than 1,000. So how is it that enzymes accomplish what they do? Well, it relates to what you actually see on the screen. Okay? This describes catalysts in general. So what I'm showing you on the screen will be true to some extent of a chemical catalyst as well. But enzymes are a very special case. Think about a reaction. A reaction has to go through what we think about as an energy barrier. If I put two molecules together, there's an energy barrier that's necessary to join those two molecules. That energy barrier is depicted here. We could think about molecule A over here and molecule B over here. And these two molecules have to bump into each other in the right orientation at the right velocity in order to join. If they hit in the wrong direction, if they hit too softly, or they hit too hard, they're not going to join together. The energy by which they join, by which they come together and they hit each other, we can think of as what we describe as the transition energy. This is the energy we have to put in to make this reaction work properly. The transition energy is the barrier of energy for those two things coming together. It's a barrier. Here's the energy of the molecules that we start with down here. Here's the energy necessary to get them to react. And after they react, they release energy. When they release energy, they end up at an energy that's lower than what they started at. That's known as a favorable reaction. A favorable reaction. Okay? Well, you can see that we're going from reactants to products. You can see that the energy change isn't big, but this energy change to get up to that transition state is pretty good sized. This is going to vary for each reaction a little bit. This is what happens if we have a reaction that is not catalyzed by an enzyme. No enzyme, no catalyst, no nothing. These two molecules bounce into each other, and they've got to bounce into each other just right in order for that reaction to occur. The reaction on the bottom compares the uncatalyzed, which you saw in the top one, with the transition energy necessary for a catalyzed reaction. You'll notice that the catalyzed reaction transition energy is a lot lower. It's a lot lower. Now, another important point. There's no difference in the energy of the reactants or the products for either. They're the same for both. Enzymes do not change the overall energy of a reaction. They can't do it. They cannot change. I'm going to repeat that. That's a very important point. They cannot change the overall energy of a reaction. The reactants start out at the same place. The energy of the products stays at the same place. What enzymes do is they change the energy of the transition state. They make it much more likely that they're going to hit and react. By lowering the transition energy, they make it much more likely that these guys are going to come together and make the product that they make. Now, we'll talk later about how they do that. If I have, well, actually, I'll tell you here. If I have A and I have B, they've got to hit in the right orientation. If they don't hit in the right orientation, they won't stick together. If they come in like this, they're going to bounce off. Enzymes grab A and B. They have binding pockets for A and B. And then they come together only in one orientation. The example I like to give is the enzyme hexokinase. You don't need to know the name of the enzyme. We'll talk about it later. Hexokinase combines a phosphate from ATP with the sugar glucose. This is the first reaction, the breakdown of glucose. What happens? The enzyme has binding sites. It has things that can hold on to these two molecules. ATP gets bound up here. Clunk. It's got it. Glucose gets bound over here in the same enzyme. Clunk. It's got it. The binding of those two things causes the enzyme to do something that only enzymes can do. It causes the enzyme to change its shape. You've already seen with hemoglobin how a slight change in shape resulted in additional oxygen binding, release of oxygen. You saw effects of slight changes in shape. The binding of ATP and glucose causes this enzyme to change shape, and the change in shape that it does is really cool, folks. The change in shape is like this. It puts them together. And it puts them together in only one orientation. It puts them together at the right energy, and it makes the reaction possible. And then once that reaction has happened, the enzyme relaxes and everybody goes home. Pretty cool. Enzymes are flexible. Proteins are flexible, right? This flexibility is what gives enzymes the ability to speed up a reaction 180 quadrillion times. And no, I'm not making that number up. Okay? And I've lost my pointer. There it is. I get so excited I forget where my pointer's at. Okay. Not everybody's excited about that, but that's okay. Okay. Questions about that? Do you understand that? Okay. All right. Well, that's how an enzyme works. That's how an enzyme works. Okay. Here's another comparison of rates. This one blows my mind as well. Okay. If I look at a reaction, and this reaction is for the breakdown of hydrogen peroxide. Okay, hydrogen peroxide is what we call a reactive oxygen species. It's kind of nasty to a lot of things in our cells. We have an enzyme called catalase that breaks it down so that if we have if we have hydrogen peroxide accumulating, then it will treat it. If you take hydrogen peroxide and you put it on your finger, have you ever done this before? You get a cut, you put hydrogen peroxide on your finger, what happens? It bubbles. Okay? What's happening is you're seeing catalase is breaking down hydrogen peroxide into oxygen and water. The bubbles are the oxygen. Okay? Bacteria don't have that enzyme, and so hydrogen peroxide kills them. So you put it on your cut to disinfect your finger. You kill the bacteria, but you don't kill your own cells because you've got protection with that enzyme. Okay? All right. What does this enzyme do? Well, it converts converts hydrogen peroxide into water and oxygen. You don't need to know that. What you need to know is the fact that if we say that the uncatalyzed rate is a 1 molecule per second, and we compare it to a platinum surface, we could make about 27,000 of those per second. And if we take an enzyme, one enzyme, that is one protein enzyme catalyst, not 50, not 100, not a million, but one of these will produce 65 million of those per second. Each enzyme will produce 65 million molecules of product per second per enzyme. That's mind-boggling. I'd like to give the analogy of an enzyme to a factory, right? We think about a factory. What does a factory do? It produces automobiles, right? That factory producing automobiles has certain limitations. It might be, do we have enough fenders? Do we have enough engines? Do we have enough supplies to make this happen? Those are the raw materials. That's what the enzyme is using. Do we have enough ATP? Do we have enough glucose? Do we have enough hydrogen peroxide? If we have enough materials, okay, then the factory is able to ultimately produce a certain number of automobiles. What's that going to be related to? The number of people working in the factory, how efficiently they're working. We're going to get to a maximum capacity of a factory. And the capacity of that factory is not going to change even if I put 10 times as many fenders out front, right? If I'm short fenders, if I'm waiting for fenders, then it's going to reduce the production of the factory. But as long as I have everything I need there, the factory is going to produce things at the same rate. Everybody understand that? I've just described a very important concept about enzymes. It's called maximum velocity. Maximum velocity. We're going to talk about several things about enzymes that are important. I've just described one of them to you. Maximum velocity. What is the maximum velocity of an enzyme? It's the same as the maximum velocity of a factory, except for it's a hell of a lot faster. What limits a factory? It's the supplies. It's the workers. What limits an enzyme? Well, there are no workers. It is the supplies. There are workers as well. What happens if I increase the number of workers catalyzing the reaction? What would you say the workers are in the reaction process? The enzymes, right? There's 65 million molecules of product per second per enzyme. Let's say I put, I don't know, 2 enzymes there. Do you suppose I'm going to double this number right here? The correct answer is yes. Yes, I will. If I make twice as many factories, won't I make twice as many cars at maximum capacity? Once I get to maximum capacity, I can't go any faster, but if I double the number of factories, I can get twice as many cars. If I double the number of enzymes, I can get twice as many enzymes, and that is a very important part of maximum velocity of an enzyme. I know seniors in biochemistry who don't know that. If you guys understand that, you've got a very important concept down. The very important concept is this. Maximum velocity depends on the amount of enzyme. Maximum velocity depends on the amount of enzyme. If I double the amount of enzyme, I will double the maximum velocity. We're going to use that piece of information in just a little bit. We call maximum velocity Vmax. Vmax. Okay. Here's a depiction of what I just told you. Okay? Here's a depiction of what I just told you. If I take, and let's imagine, how do I do an experiment where I might measure Vmax? Here's how I would do it. And by the way, I commonly ask students how I do experiments on exams. How would I do this experiment? Here's how I would do it. When I do an experiment, I only want to have one thing that varies. If I have 6 things varying, I can't tell which variable affects the result I get. So when I do an experiment, I want to have one thing that I vary. I'm going to start with 20 test tubes. I want to see how many molecules of product I can make per second with a given enzyme. And yes, that number will vary from one enzyme to the next. I want to know for my particular enzyme how many molecules of product I'm going to make. I take 20 tubes. Into each tube, I put the same amount of buffer. Into each tube, I put the same amount of enzyme. And into each tube, I put a different amount of substrate. Substrate is the molecule the enzyme acts on. It could be more than one molecule, it could be 2, could be 3. But the substrate is what the enzyme is catalyzing the reaction on. Tube 0 would have no substrate. Tube 1 would have some amount. Tube 2 might have 2 or 3 times this amount. Tube 3 might have 4 or 5 times the amount, etc., etc. So I go further along. Each tube gets more and more and more substrate. Okay? The only variable here is what? Substrate. The only thing that's varying is the substrate. Okay? I let every tube react for the same amount of time. So time is not a variable. And I ask the question, how many molecules of product do I see in each tube? So I can ask the question, how does the amount of product vary with the amount of substrate that I have? Okay? If I have 0 substrate, how much product am I going to make? If I have a tiny amount of substrate, I'm going to make a tiny amount of product. This is a measure of what's called velocity on the y-axis versus substrate concentration on the x. What the hell is velocity? Velocity is very simple. If I say velocity of a car, you would say, well, 30 miles per hour, right? If I say velocity of a reaction, it's the concentration of product over time. Velocity is the concentration of product over time. If I make a lot of product, I'll have a high concentration. If I make a little bit of product, I'll have a low concentration. If I do a reaction in tube number 1 where I have a very tiny amount of substrate, I'm going to make a very tiny amount of product. Why is that? Because for the same reason, if I don't have enough fenders for the factory is going to wait around until a fender shows up before it can finish a car, right? The enzyme is sitting here spinning its wheels, waiting for a molecule of substrate because there's not very many molecules of substrate in that reaction, in the tube, right? Not very many molecules of substrate in the tube. However, if I go further and further and I measure tubes that have more and more substrate, it's less likely the enzyme is going to be sitting around waiting for substrate. That means it's going to be making more and more product. The more substrate I add, the more product will be made. That's pretty cool. But then something odd happens. This doesn't just go up linearly. You might say, well, I add more substrate, I get more product. So what? Why does it start flattening off? I'm reaching maximum velocity, and why do I reach a maximum velocity at all? Okay? For the same reason that the factory reached the maximum velocity. It's limited by the number of workers I have there and the amount of materials that they have. Materials over here are in high abundance. But now, I don't have enough workers. What I've just done is I've saturated the enzyme with substrate. Important concept. I've saturated the enzyme with substrate. That means that the enzyme is not waiting for substrate at any time. Saturation means that. The enzyme always has substrate waiting to get in there. What I'm limited in are the number of molecules of enzyme that I have. This is Vmax. If I were to double the number of enzymes that I had, what do you suppose would happen to Vmax? Double. This would be way up here instead of way down here. Vmax varies with the enzyme. Now, a lot of people don't know that. A lot of people think Vmax is a constant for an enzyme. As they say, I know seniors that think that. That's not true. What they're thinking of is a related idea. The related idea is something called kcat, K with a lower subscript of CAT. Kcat is also called the turnover number. Kcat, we know if we say that Vmax varies with the amount of enzyme, then we can't compare Vmax of one enzyme with the Vmax of another enzyme because it depends on how many enzymes I used, right? What if I take the enzymes out of the equation? What if I said, here's the total number of products I had, and here's the total number of enzymes I used, and I divided the products by the enzymes? Now the enzymes wouldn't factor into the equation. I'd know how many I got for each enzyme, right? That was what we had when we had the 65 million molecules of product per molecule of enzyme. I had taken the enzymes out of the equation. Kcat is related to Vmax. Kcat is equal to Vmax divided by the concentration of enzyme. Kcat is equal to Vmax divided by the concentration of enzyme. What does that give me? It gives me a number. It gives me something we call the turnover number. And that number is the number of molecules of product per molecule of enzyme per time, in this case, second. Now, I'm throwing a lot of things at you today. I know that these are a little mathematical. And I'm going to give you some time to digest that. But before yeah, there's a question right there. Miriam? Yeah. So, Kcat, how do I calculate Kcat first? I calculate Kcat by taking Vmax and dividing it by the concentration of the enzyme that I used. That gives me a number. And that number is equivalent to the number of molecules of product per molecule of enzyme per second. So just a second. What that means when I've got catalase and I say 65 million per second, that means that every molecule of enzyme is producing 65 million molecules of product every second. That's mind-boggling, folks. It tells us that the nanoscopic world of molecules is very different than the macroscopic world that we see out here. You hear people talk about nanotechnology and the excitement of nanotechnology. It's partly based on what I just told you right here. The nanoscopic world is different than the macroscopic world. Had a question back here.
Yeah, so if Vmax is not constant, how do you find that number?
Okay, if Vmax is not a constant, very good question. How do I find that number? All right, I do it by the way I just described, which is I take, say, 20 tubes, varying substrate, but with a constant amount of enzyme. So for every reaction that I just described, I have the same amount of enzyme that's there. So the amount of enzyme is not a variable. That concentration of enzyme I use is what I plug into that equation. Remember the equation that said Kcat equals Vmax divided by the concentration of enzyme? That concentration of enzyme I used in that reaction is just what I just said. Does that make sense? Yeah? Yes, that's a good question. Does an enzyme ever slow down, break down, or any of that? Enzymes turn out to be pretty, it varies from enzyme to enzyme. Some enzymes are very, very unstable. They don't last a long time. They're probably better in cells than they are when we put them in test tubes. We don't exactly know what protects them in a cell, but so we may not have that in a test tube. Other enzymes, you can beat on them with a hammer, and they still do things. Ribonuclease that I talked about the other day is an example of an enzyme that you can do almost anything you want to it, and ribonuclease will continue to catalyze a reaction. So it varies from one enzyme to another a lot. Okay, I'm going to introduce another concept, and then we're going to sing our song for the day. Okay? All right. The other concept I want to introduce to you is something called, and yes, I will go through this again on Monday. Okay, so I understand this is a little complicated. I'm throwing a lot of things at you. Okay? The last concept of the day is something called Km. Km. You can relate Vmax and you can relate Kcat to something equivalent to velocity. How fast is a reaction going? Right? How fast is this enzyme working? How fast is this reaction going? There's other things that we could imagine about an enzyme that we might like to know. And one of those things we might like to know about an enzyme is how much does an enzyme like to grab onto the molecule that it catalyzes a reaction on? It's called the affinity. A-F-F-I-N-I-T-Y. What is the affinity of an enzyme for its substrate? An enzyme that has a high affinity for its substrate loves to grab its substrate. An enzyme that has a low affinity for its substrate doesn't. And how can we tell the difference between the two? Well, one of the ways we can tell is by how much product it makes over time. Okay? And how much concentration plays a role in that. It's depicted here. Here's that V versus S that we did, velocity versus substrate concentration. There's Vmax that we determined. And if we want to measure the affinity of an enzyme for its substrate, we have to go to a quantity known as Vmax over 2. Take the measure of Vmax, divide it in half. And then ask the question, what substrate concentration gives that? This is the substrate concentration that gives Vmax over 2. And it turns out we call that Km. Now, first, I'm going to tell you something that a third of you are going to tell me on the first exam completely wrong. So I'll tell you what your wrong answer is going to be. Even after I've told you, a third of you are going to be wrong. But I'll tell you in hopes that you listen and you don't get it wrong. On an exam, I'm going to ask you in one way or another to tell me what Km is. And a third of you are going to tell me Km is Vmax over 2. Even though I tell you that's not the right answer, a third of you are going to tell me in one way or another that Km is equal to Vmax over 2. And that'll be wrong. Km is the substrate concentration that gives Vmax over 2. Km is a substrate concentration. It is not a velocity. It is a substrate concentration. It is the substrate concentration that gives Vmax over 2. Okay. Now I'm going to come back. I'm going to repeat that for you on Monday. I've got a song. The song covers a couple things I haven't talked about today, but I think you'll get the idea from the song. It's called Enzymes. It's to the tune of an old 60s song. You may not know the tune, but if you do, please sing loudly because you can drown me out. Reactions alone could starve your cells to the bone. Thank God we all produce enzymes. Units arranged to make the chemicals change because you always use enzymes. Sometimes mechanisms run like they are at the races. Witness the Kcat of the carbonic anhydrases. How do they work? Inside of the active site, it just grabs onto a substrate and squeezes it tight in an enzyme. catalysis in an enzyme. V versus S in an enzyme. All of this working for you. Enzyme, enzyme. Energy peaks through what an enzyme defeats in its catalysis. Enzymes. Transition state is what an enzyme does great, and you should all know this. Enzymes. Catalytic action won't run wild, don't get hysteric. Cells can throttle pathways with an enzyme allosteric. You know it's true. So when an effector fits, it will just rearrange all the subunits inside an enzyme. Flipping from R to T, enzyme. Slow catalytically, enzyme. No change in delta G. Enzyme, enzyme. You should relax when seeking out the Vmax, though there are many steps. Enzymes. Lineweaver-Burke can save a scientist work with just 2 intercepts. Enzymes. Plotting all the data from kinetic exploration lets you match a line into a best-fitting equation. Here's what you do. Both axes are inverted, then you can determine Vmax and establish Km for your enzymes. Sterically holding tight, enzymes. Substrates positioned right, enzymes. Inside the active site, enzymes, enzymes. Okay, see you guys on Monday.